Today, we're diving into a scientific detective story about a killer that breaks all the rules of biology. It's not a virus, it's not bacteria, and weirdly enough, it might already be inside you. Let's get into it. For centuries, we had a pretty solid idea of what an infection was. It had to be something alive, right? Something with genes, like a virus or a bacterium. But what if it wasn't? What if the real killer was something far simpler? Something that's already a part of all of us, just twisted into the wrong shape. So what you're looking at here are nerve cells from a mouse, and if you see those bright red clumps, that's our culprit, right there in the act. That's not some foreign invader. It's an accumulation of a rogue protein, and it is literally eating away at the brain from the inside out. So how in the world did we even figure this out? Well, the whole story starts with a medical mystery that, for the longest time, seemed completely impossible to solve. For decades, scientists were just stumped. They were looking at these fatal brain-wasting diseases, like scrapie in sheep, or a really strange human disease called kuru. It was obvious they were infectious, they could spread, but nobody could find a microbe. And the infectious agent, whatever it was unbelievably tough. It could survive radiation and chemicals that would destroy any virus or bacteria we know of. It just didn't make any sense. And the answer, when it finally came, was absolutely revolutionary. The culprit wasn't a living thing at all. It was just a protein. So back in 1982, a neurologist named Stanley Prusiner came up with this really radical idea. He coined the term prion. He basically mashed together the words proteinaceous and infectious to get prion. And his argument was that this infectious agent was nothing more than one of our own proteins, just misfolded, a protein gone bad. This image right here tells the whole story. See, we all have this normal, totally harmless protein called PRPC on the surface of our cells, but if it bumps into the misfolded prion form, PRPSC, it transforms. Just look at that dramatic change in shape. It's the exact same chain of amino acids, but it's folded into a completely different and dangerously stable structure. And this is the absolute key. The normal protein has this soft helical structure, kind of like a spring. Our body can break it down and recycle it no problem. But the rogue prion, it refolds into these rigid, flat sheets. This new shape makes it incredibly stable. It's insoluble. The body's enzymes can't break it down. For all intents and purposes, it's indestructible. Okay, so how does one little misfolded protein cause a fatal disease? Well, the answer is a terrifyingly efficient chain reaction. So how does it, replicate? It's not like a virus, which copies its DNA. No, instead, as this model shows, the prion acts like a dangerous template, or maybe a seed, that kicks off a devastating domino effect. It's like a three-step nightmare. It's so simple. First, an infectious prion makes contact with one of its normal, healthy counterparts. Then, it physically forces that healthy protein to snap into its own wrong shape. And just like that, now you have two prions. Those two then find two more healthy proteins and convert them. Then 4 become 8, and on it goes, an exponential cascade of misfolding. As this chain reaction snowballs, all those sticky, misfolded prions start clumping together. They form these long, tough fibers called amyloid plaques. These are the red clumps we saw in that first image. It's essentially protein garbage that the brain has absolutely no way of cleaning up, and it just starts to accumulate. And the result of that buildup is just devastating. The plaques kill neurons. This quote here really describes it perfectly. The brain tissue develops actual holes, creating a spongy architecture. The medical name for this is spongiform encephalopathy, and it's every bit as destructive as it sounds. So let's connect this process to the real world. What do these diseases actually look like? The list of prion diseases is pretty chilling. We've got scrapie in sheep, bovine spongiform encephalopathy, you've probably heard of that one as mad cow disease, and chronic wasting disease, which is a huge problem in deer and elk populations. And in humans, you have the infamous Creutzfeldt-Jakob disease, kuru, and even a condition with a terrifyingly literal name, fatal insomnia. And they all share the same grim playbook. They are relentlessly progressive. They can relentlessly lie dormant in the body for decades. The incubation period can be 5, 10, even over 20 years. But once the symptoms finally appear, the decline is terrifyingly fast. And right now, we have no effective treatment. They are, without a single exception, fatal. So why does this one super rare, very strange protein matter so much to all of us? Well, because the way it works has completely changed how we think about a whole host of other brain diseases. Discovering the prion gave scientists a whole new mechanism for disease. It created a new way of thinking, the prion paradigm. It proved that information could be transmitted not just by genes, but by shape alone. A simple protein could become infectious just by folding the wrong way, and then teaching other proteins to do the same. And this is why it is so critically important. Scientists now believe that many far more common neurodegenerative diseases work in a very similar prion-like way. Diseases like Alzheimer's, Parkinson's, ALS, Huntington's. In all of them, we see other kinds of proteins that misfold, clump together, and spread damage through the brain. The prion literally provided the deadly blueprint. And that leaves us with this final, really profound question. The prion taught us that a simple protein could turn into a relentless killer just by changing its shape. So you have to wonder, how many other diseases that currently baffle us are quietly following that very same deadly blueprint?